This is the second video presentation for the CASE Animal and Plant Biotechnology course. As you view this video, be sure to make notes of important information. Feel free to pause, repeat, and resume the video whenever necessary. Plasmids and Bacteria, Lesson 2.2, Genetic Transformers. You completed Activity 2.2.1, Transformation Preparation, during which you prepared media to use during bacterial transformation throughout the next few activities. This presentation provides an overview of the process of transformation. It introduces plasmids and how they are utilized in genetic transformation. Transformation is one method of modern genetic modification. A transgenic organism has a genetic makeup containing DNA which was physically transferred from another organism. Examples of transformed organisms include oil-eating microbes, pest-resistant crops, and pharmaceuticals from biofarming practices. Plasmids are extrachromosomal DNA arranged in a circular shape. Plasmid DNA does not interfere with the host chromosome functions. Restriction enzymes are used to cut plasmids. A single cut will open a plasmid molecule, making way for the insertion of new genetic material. Once a plasmid molecule is closed, it will replicate within the host. A bacteria takes up small pieces of DNA. The plasmid DNA may be expressed. During the next few activities, you will attempt to transform bacteria with the PGLO plasmid. The PGLO plasmid has two major functions: bioluminescence and ampicillin resistance. Bioluminescence, or the glowing characteristic, is a result of the addition of the green fluorescent protein, or GFP. In order to activate the GFP, arabinose is required. Bioluminescence indicates whether transformation has taken place. The E. coli strain HB101 is a benign bacterial strain, not to be confused with the more dangerous strains. E. coli HB101 is widely used in research. Non-transformed E. coli HB101 will not survive in the presence of ampicillin, a common antibacterial agent. In the media you prepared, you have various combinations of agar, ampicillin, PGLO plasmids, and arabinose. Before you move on, take a moment to predict what will happen in each plate if bacterial transformation has taken place. Will bacterial colonies form? Will the colonies exhibit GFP? Pause the video to record predictions for all 4 plates in your notes. Include notes regarding why you believe each outcome will happen. Resume the video to compare your predictions with those of experts. The first plate contains LB agar and no PGLO genetic material. There are no genetic modifications made to the culture on this plate. Anticipated results include heavy bacterial growth since LB agar is conducive to the growth of E. coli HB101. Because no PGLO genes are present, the bacteria should not exhibit GFP. The second plate contains LB agar, ampicillin, and no PGLO genetic material. No modification should take place on this plate. No growth is anticipated as this sample contains ampicillin, which inhibits the growth of E. coli HB101. The third plate contains LB agar, ampicillin, and PGLO genetic material. Genetic modification should take place, but only a limited number of bacterial colonies will appear, as not all will take up the plasmid to be resistant to ampicillin. Although the modification will take place, the colonies will not exhibit bioluminescence as there is no activator. The final plate contains all treatments. The presence of LB agar, ampicillin, arabinose, and PGLO genetic material should result in a limited growth of bacteria that glow under UV light. In short, the plates with PGLO and ampicillin should express limited growth of bacteria. Only the plate with arabinose, used to activate the GFP, should glow. As a review, mark or highlight 3 key points in your notes that are important to remember. List 2 ideas or concepts that relate to previous knowledge. List any questions you have about this topic. Discuss these with your instructor. Keep any notes you have from this presentation organized and available for use throughout the course. Through Activity 2.2.2, Glowing Genes, you will transform bacterial cells to exhibit bioluminescence and ampicillin resistance in order to understand how plasmids are used to insert genetic material into bacteria. Your teacher will provide more instructions.